Journey into memory. Where do memories reside? Our brains are intricate webs of billions of neurons, tiny messengers that communicate through electrical and chemical signals. For decades, scientists believed that memories were formed and stored solely at the synapse, the junction between two neurons. Imagine them as tiny bridges connecting thoughts and experiences. The strengthening or weakening of these synaptic connections, a process called synaptic plasticity, was thought to be the primary mechanism behind learning and memory. Picture a well-trodden path in the woods becoming more defined with each step. However, recent discoveries have unveiled a hidden layer of complexity within the neuron itself, revealing that the nucleus, the very heart of the cell, plays a critical role in memory formation. This is where epigenetics takes center stage. Beyond the synapse, the epigenetic symphony. While synaptic plasticity remains crucial, we now understand that it's only one part of the memory story. Imagine a grand orchestra playing a symphony. Synaptic plasticity is the melody, but the harmony, the richness, and the nuances of the music come from the epigenetic layer. Epigenetics refers to changes in gene activity that occur without altering the underlying DNA sequence. Think of it like this. If our DNA is the alphabet, epigenetics is the grammar and punctuation, dictating how the letters are arranged to convey meaning. This epigenetic layer adds another dimension to our understanding of memory formation, revealing a dynamic interplay between genes and the environment. Just as our experiences shape our memories, they also leave their mark on our genes, influencing how they are expressed. The epigenetic code. Whispers on the DNA helix. Imagine our DNA as a long, winding ladder. Attached to the rungs of this ladder are chemical tags called epigenetic marks. These marks act like switches, turning genes on or off, or fine-tuning their activity levels. One key player in this epigenetic orchestra is chromatin, the complex of DNA and proteins that makes up our chromosomes. The way DNA is packaged within chromatin can either expose or hide genes, influencing their accessibility to the cellular machinery that reads and translates them. This means that even though our DNA sequence remains largely unchanged throughout our lives, our epigenome, the overall pattern of epigenetic marks, is constantly being shaped by our experiences, diet, and environment. A mouse, a maze, and the memory molecule. Enter Johannes Gräff and his team of researchers at the EPFL in Switzerland. They embarked on a groundbreaking journey to explore the role of epigenetics in memory formation using a clever experiment involving mice. They trained mice to navigate a maze, a task that requires learning and memory. As the mice learned the maze, the researchers observed changes not only in synaptic connections, but also in the epigenetic landscape of specific neurons within the brain regions associated with memory. Specifically, they focused on a molecule called NPAS4, known to be involved in neuronal plasticity. They discovered that blocking NPAS4 in the hippocampus, a brain region crucial for memory, impaired the mice's ability to form new memories of the maze. Rewriting memories, epigenetics in action. The implications of this finding are profound. It suggests that NPAS4 acts as a molecular scribe, translating experiences into long-lasting epigenetic marks on our DNA, thereby shaping the very fabric of our memories. Think of it like this. NPAS4 acts like a diligent librarian, carefully cataloging and shelving books, memories, in the vast library of our brains. By altering the epigenetic landscape, NPAS4 ensures that these memories are stored and retrieved efficiently. This study provided compelling evidence that epigenetic modifications, particularly those orchestrated by NPAS4, play a crucial role in memory encoding. It's no longer just about the strength of the connections between neurons, but also about the internal state of those neurons, governed by their epigenome. From mice to mankind. A future of enhanced memory. While these findings primarily stem from research on mice, they open up exciting new avenues for understanding human memory and its disorders. Imagine the possibilities if we could manipulate our epigenome to enhance memory formation or even erase traumatic memories. Scientists are particularly interested in exploring the potential of epigenetic therapies for treating cognitive disorders like Alzheimer's disease, where memory loss is a hallmark symptom. By targeting epigenetic mechanisms, researchers hope to develop novel treatments that could slow down or even reverse cognitive decline. The journey into the epigenetic world of memory formation has just begun. As we delve deeper into this fascinating realm, we unlock new secrets about the workings of our brains and unveil the intricate dance between nature and nurture in shaping who we are.